little bit about nature metrics at the end. But this talk is going to have 4 parts to it. And just to give you a little preview of the last part, here's a really great tweet by Ben Evans, who's a tech industry observer. And I'll explain why I think this is such an interesting piece of wisdom. So as Vera said, I'm a faculty member at the University of East Anglia, and it's a university. It's also famously the New Avengers facility, which is great because we have neighbors who understand the importance of field work and high tech, and we get along very well with them. I thought also because I'm told that there's some students in here, I've been told that it's a good idea to give a little personal background for students to see how our careers have progressed a little bit. And so I'll give you a little bit of my own personal background. I'm Chinese American, living in the UK, lots of military and academics in our family. I'm a suburban city boy. I used to work in the IBM building in New York City, my first job, and that was next to Trump Tower. So I've been here next to him for a long time. And then about 3 years into my corporate career, I decided I really didn't like it. And I thought tropical forest to be a lot like Manhattan, and it turns out to be true. You basically just look up all the time. I split my time between China and the UK, well, pre-pandemic. I made a decision early on, which you may or may not decide is a good idea for you, to change my study subject every 5 years or so. So I worked on ant plants, ecotourism, economics, monkey hunting, ethnobotany, game theory, and so on. And now I'm really, these days, I discovered after all these different subjects that what I'm really interested in is management, how to get someone to do something. And that's true in biology and also true in conservation. As Vera's pointed out, as part of that, I co-founded this company, Nature Metrics, which I'll talk a little bit about. Okay, so I'm going to give some very brief background of what about DNA barcoding and what they are, and then I'll go into some applications. So DNA barcoding is just a short sequence of DNA. The most famous one is this region of the cytochrome oxidase 1 gene. It's about 650 base pairs long. If you sequence this from an insect, you can take that sequence, stick it into this website here, BLAST, and you get back a series of sequences that it matches. And ideally, it'll look something like this. It'll really matches Anopheles quadrimaculatus, and it doesn't really match the other Anopheles. And so you can conclude that this sequence comes from a mosquito and of that species. And the thing is, what happens when you have many insects in a single sample, what you could do is metabarcoding. So you take your insect soup and you extract the DNA. In many of the pipelines, you do PCR amplification, and then you send it off to the Illumina sequencer. And what you get back is just many sequences of DNA, most of which are copies. So then you reduce them down to clusters of reads. And the idea is that each cluster, if you did it correctly, represents one species. And since it's a hypothesis, we call it operational taxonomic unit. And then you do some taxonomic assignment and try to identify what species is each sequence. You do this for lots of samples. So in the end, your output is a standard output that we see in ecology for decades, which is a sample by species table. The difference, though, is that it's automatic. You get many species if you do it in particular ways, to use the right primers and so on. And I think this is more important. It's easy to add samples, and that changes, I think, things even more than adding species, because we can sample. You can take many samples. You're not worried about having to identify all the species in those samples, and then you can take kinds of samples that are really easy, like water. And so early on, when we were thinking about doing this, we started working with people who are actually doing real biodiversity studies morphologically. And we worked with a group working in China, and they took moth transects up and down a mountain and did this, pinned them and then took each moth and then matched it to moths. And they ended up with some hundreds of moth morphospecies. And we said, well, do you have any extra moths? You could give them to us. We could just turn them into moth soup. And they said, sure, no problem, right? And they ignored all the micromoths, whereas we did everything in the moth soup. We just took half a bottle from each one. And it took us half the time for them than they did. And we did everything in that soup. And what we found was we compared it, and they were able to show, for example, that two samples that were from the same altitude had similar compositions, and if they're from different altitudes, they have different compositions. And we showed the same thing using ours, but using more species. So we were able to recover what they were doing. This is using an old sequencer. And so then we started immediately thinking, what can we do with these large sample by species tables? And one of the first questions we had was, how do we know if a nature reserve is effective, right? This is a fundamental question in conservation biology. And given what's going on today, China and Vietnam have banned the wildlife trade in their country, and we want to know, does that actually have an effect? Will that change behavior upstream to hunting? And so is it possible to see a positive effect on conservation, on wildlife populations as a result of these bans? So you have to take some baseline data. And it turns out last year, conveniently, Maxwell et al. Published a review of area-based conservation, including nature reserves. And a fundamental question is, do the world's nature reserves have positive biodiversity outcomes? They reviewed the literature and they concluded that we do not know. This is an amazing conclusion, right? That after all this time, we do not know whether or not nature reserves are effective. And it's not too surprising. Here's a nature reserve taken by my friend Andrew Tilker, and this is a rainforest from Vietnam. And there are two medium-sized or large mammal species in this picture. And if you imagine walking around here and trying not to trip and trying to keep everything going, how will you see those mammals? Well, it turns out that they're right in front of you. There's one right here and there's one right there. Those are humans. So that's the large or medium-sized mammal. So you can't even see necessarily your own team members. Well, so we thought, well, what can you do? So there's a nature reserve just south of Kunming, where I work, and it's about the size of Singapore, and it's just the top of a ridge of mountains, and so very hard to survey. And we'd like to know where the animals are and where are they in that nature reserve, and which species are there. And you could put in camera traps, you could do surveys. But there is kind of a shortcut, we thought, which was to collect leeches, because there are lots of leeches during the rainy season there. And importantly, we can get people to collect them for us. So we got 163 forest rangers, paid them 300 RMB, which is about 40 pounds, or maybe 50 euros. And then in about 2 months, they collected 30,000 leeches, which was much more than we expected. And over 172 ranger patrol areas, right? So they go in and do a walk and then just picked up any leeches that they found and gave them to us. So we choked on that for a while in our lab. And then we finally did some metabarcoding on that after we figured out how to extract DNA in that way. We actually ended up renting empty apartments nearby to be able to do clean DNA extractions. And we got amphibians, birds, mammals, and a little bit of reptile DNA. Now we're able to pull it out. Large and small, and so on. And so you can see that I'm not going to go into any detail here, but we gave them many tubes. And so those tubes automatically cause them to replicate their sampling. So one tube fills up, they go to another tube, they go to another tube. So one single walk meant that a single ranger was taking multiple samples, replicate samples in their walk, right? It's not a perfect replicate, but it's pretty good. And we didn't have to go back. That was key. We did some bioinformatics and so on, and took a long time to write all this out. And in particular, I just want to point out that Sumervo et al. Wrote a really important piece of software called ProTax, which we use to do taxonomic assignments. And then started applying what's called a multi-species occupancy model, right? And a multi-species occupancy model separates two processes in ecology. First of all, it separates whether or not a species is present in a site in reality, right? So that depends on things like the environmental condition of that site. And so in our case, we just asked, does elevation determine any part of whether a species is present or not? And also, does distance to the edge of the reserve determine anything about whether a species is present or not? But even if a species is present, it's possible that you don't detect it. So you don't see it, or it doesn't get eaten by a leech. You don't pick up that leech, or you fail to pick it up in the bioinformatic process. And so there's a detection process. So this becomes a hierarchical model of multiple species. And in our case, we have one covariant, which is we have a number of leeches in a sample. And so presumably more, and we do find more leeches, the more species that you get, so the higher detection. And so that is the multiple species hierarchical model. And it's been really tough to get this thing to fit. But you can see here that these are the places where we successfully got samples from. And this is the interpolated map from after the multi-species occupancy model. And you can see that we estimate that the highest species diversity is in the southern region, which is more rainy and better forest cover, and also bigger and higher. And so I'll just give you some individual maps. So this is the muntjac. Muntjacs are resistant to hunting pressure, and so they tend to be found in lower elevations. And this is a leopard cat, and it tends to be found in higher elevations. And we can ask, overall the species, what kind of effects do we have? And you can see that both mammals and birds, which is this group here, and amphibians and reptiles, which is that group there, they both show a general group-wise increase in occupancy. That's probability of being there with elevation. And then individually, you can see a lot of those increases. And then you can also see a few that are negative. We see a much weaker effect from distance to reserve edge. Not surprising, I think, because there's a very thin reserve. And most of the elevation and distance are correlated with each other. So which species are showing that positive effect? Which species are showing the negative effect? Well, there are 5 species that show a negative and strongly negative effect. They found more often in low elevation. 3 of them are domesticated species: sheep, goat, cow, and goat. We know that from camera trapping and just general knowledge that in fact people do bring their domesticated animals to graze in the lower parts of the park. There's the red muntjac we saw before, and a rat. All right, and these are species. This is a species that's resistant to hunting, and we know that there is some hunting in this area. So that's a camera trap photo on the left. And then everything else either does not show any effective elevation. A lot of frogs this way, and quite a few that are large mammals and so on show this effect if there's enough sample size. So, question: does this nature reserve have a positive biodiversity outcome? I'd say yes. We are finding the species that we would like to find in that park, but there's much room for improvement. There's literal room for improvement in the lower elevation areas. And then the key, I think, is to understand that these data have some useful management properties, right? So we did another big survey of leeches a couple of years later. It was 2 more months to get a bunch of leeches. It's something that you can just do over and over again over the entire park, right? So it's efficient and repeatable. The laboratory isn't so efficient, but the collection is very efficient. It's direct, right? We're directly measuring biodiversity. We're not measuring some proxy of biodiversity, and it's granular. We're measuring individual species in individual places, and it's understandable. I can show a map of individual species. I can show a map of all the species stacked on top of each other, and so it's auditable. You could then say, well, we think there are bears in this area. You could go check if not. So, and then that means that you can then do what's called condition incentives. You can say, I will promote you if we see an increase in the wild animal occupancy and so on. And then that becomes a management incentive. Never really been possible before to pay on outcome, but now it's theoretically possible. I'm not certain that it's possible here, but I think this kind of approach makes it possible. Okay, so we have a bioarchive paper. We've been working on the stats for quite a long time, and then our co-author Chris Baker, who's exceedingly smart, has done a very smart thing, which is to have twins. So we might see the paper come out in 5 years from now. I'm not really sure, but anyways, it's on preprint. And so these are the 2 key authors who worked on the laboratory bioinformatic pipeline and the multi-species occupancy model. Okay, so that gave us the idea then to try to do really large biodiversity surveys in other places. And so we went to somewhere also very exotic, which is England. And this is, there are no blood-sucking leeches at least terrestrial blood-sucking leeches in this part of England. They're all in London. And so we did here instead was there's a lot of semi-natural habitat, but we wanted to somehow sample that, and it's mostly private, lots of domestic animals, right? And so it's very hard to get access to do sampling of any kind, and it's a huge area, right? And so what we did was we took advantage of something that happens in the home building industry. If you traditionally, correctly, if you build homes and you're within 500 meters of a pond in the UK or in England, you have to survey it for this one protected species, the great crested newt. And for a long time, it was 4 people. One person would go there, visit 4 times, and finally write a report. Now they pay a specialist company to collect some water. The water then is sent for quantitative PCR, and then you can detect or not great crested newt. And so that happens in just a few days. And so they collect lots and lots of great crested newt. Now, that's the first stage. But then what happened was Nature Space Partnership is a private company, had this very good idea that what they would do is they would systematically survey 600 ponds across this area. Not for any particular building project, just to build a map of the great crested newt. And they worked with the Freshwater Habitats Trust, Nature Metrics, and so on. And then what they did was they built that map, and then they divided it into 4 zones. So the red zone is high occupancy, great crested newt. Amber is less, green is less, and then white is there's no great crested newt here. And then if you're building some buildings, you do not have to survey the pond. What you do is you say, okay, am I the green, white, green, amber, or red zone? And then I just pay some money. And that money goes to pay for new ponds. So now, after all this time, well over 1,000 new ponds have been created in England as a result of this money that goes in. And the money is not spent on surveys, but actually spent on habitat creation. So this is a whole new, different kind of way of doing conservation. We estimate that the cost of this 600 ponds, that we got access to the samples afterward because they stored them, is something like $200,000 worth of sampling, even if we could do it. And there's a lot of people on telephones asking private landowners for access to those ponds. Okay, so here's my student Taiwan, and so she metabarcoded and successfully metabarcoded about 300 of those ponds. This is like the last little bits of DNA left over after the great crested newt work was done with them, and got a range of vertebrates, including the great crested newt, which is useful. And then we used a different kind of species distribution modeling method called joint species distribution modeling. And this is by Max Pickler and Florian Hartig at University of Regensburg. We built a model asking, is it possible to predict these distributions using local pond measurements, which the ecologists are taking when they collect, and also some landscape variables. What's the land cover within 500 meters of the pond, and then location. And it does okay. So I should say that this is a test metric, a performance metric called AUC. 1 means that it's a perfect prediction. So we fit a model, and then we have some data that we hold outside of the model. We don't do anything with it. And then once we have a fitted model, we say, does this model correctly predict this test data set? And so each different species are predicted different amounts, but some of the ones are predicted almost perfectly. Some of the ones are predicted okay. Some of the ones are only predicted sort of at random. So no better than random, which is 0.5 AUC, or even slightly worse, or quite a bit worse than random. So, but in general, as you can see, that most of the species are predicted sort of with about 0.7, which is usually the threshold for considering that this is a decent prediction to work with. So that's not bad, considering that we're working with absolute tiny amounts of DNA left over, and also we only have a few environmental covariates. The worst predicted species are the domesticated animals. Makes total sense, right? Pigs, chickens, pheasants, which are domesticated here, dogs. They're not determined by things like pond location. Now, what can you do with this? Well, it turns out, again, there's another paper out of IDIV and led by Jonathan Chase, and they suggest that if you use joint species distribution modeling, you can get a view of the internal structure of meta-communities. And these ponds are a meta-community. Each pond is a community, and then you have a bunch of ponds. And the idea about meta-communities is that you have three main things that determine when a species is there or not there, right? So one is that it's environmental conditions. The other one is that maybe there's a species interaction that determines if a species is there or not. And the third is just dispersal. And it looks like we can get all three of these pieces, just like they suggested. So for example, for each of the species, I'm just showing you the fish and the amphibians here. We find that some of the fish show a significant preference for larger ponds. Some of the same fish show a significant preference for ponds that were low risk of drying, which is what the ecologist goes there and just puts down a number what they think the risk of drying is. Nobody believed that this number was good. It turns out it doesn't seem so bad. And then some of the fish prefer ponds with low amounts of macrophyte cover, low amounts of water vegetation. We think this is caused by the fish, though, not that it determines the fish. And then we look at the amphibians, and we can see that two of the species, the smooth newt and the great crested newt, they like smaller ponds, not bigger ponds. They like ponds that dry occasionally. That makes total sense. And they like ponds that have high macrophyte cover. Basically, fish and these two newts don't like each other, which has always been thought to be true, and we see it in our data set. We can ask about the land cover around it, and we find that the common toad, palmate newt, and common frog, they are found more often when there's woodland around the pond. We don't see this for the great crested newt. And then some of the fish tend to stay away from when grassland is nearby as the dominant land cover. And so we have some landscape variables that seem to work. So that's the first, environmental filtering. There's also very skeptically, I think, these days with joint species distribution modeling, the possibility that you might be able to see some effective biological interactions, species interactions. So I'm quite skeptical of this too, but I'll just point out that in this case, most of the species are positively correlated with each other. So these are the correlations between species left over after you've taken all the environmental variables that you know about out of the equation. But there are two species, which is great crested newt, smooth newt, and they show a lot of blue dots here. And those are negative correlations. They are negatively correlated with the fish species. So again, we see that these newt species and these fish species are not found in the same ponds, even after you have corrected for pond size, risk of drying, and so on. So that makes sense. There's fish predation on newts. There's even a tiny little bit of newt predation on tiny fish. But this looks to me like consistent with maybe a biological interaction, predation interaction. And then finally, the last one, of course, is this idea that dispersal determines where species are. And so you can generate these kinds of plots, these ternary diagrams and JSDMs. And so this group of species in the red box, those are species that are found in ponds near each other, but the ponds are very different from each other. So if the ponds are very different from each other, they don't show a strong filtering effect. They don't show a strong niche definition, but they were in one and then they just happen to be found in the next one. Well, what species are those? They make sense. A lot of them are blue dots, and these are birds, non-waterfowl birds, some mammals, and so on. So these are the species exactly who would be using the ponds not as habitat, but more like they're using the ponds as toilets and drinking fountains and so on. So you would expect that kind of structure from them. Okay, so the thing is, the construction industry collects thousands of these ponds per year to do the work, and then this nature space thing is also spreading across all of England. The government does it too through Natural England, which is the Wildlife and Parks Authority here. And so there are thousands and thousands of ponds being collected for eDNA. And so now the country would like to do biodiversity offset. They would like to do natural capital reporting. They would like to do systematic conservation planning. Here's a way to get highly resolved biodiversity data paid for by industry. But they just have to, make sure that that happens. But the structure is set up to do this. Okay. Topic 3. How will biodiversity respond to climate change? So if you work on this, and this is not something that I've done a lot of work in, but got involved in this project, the way you can the most frequently used way of predicting how biodiversity respond is to have a climate envelope now, and then a future climate that from predictions, and then to have a species distribution model, and then have that species distribution move with the climate envelope. This is known as the climate envelope correlative approach. But the problem is everybody who works on this, and people much smarter than I am, they know that this is just one of the determinants of species distributions. And they also say that species interactions are quite important. But it's just really, really hard to know what species interactions are happening, right? And it's just more or less ignored for most cases. Okay, so tied with this is the famous Crefeld data set, which I'm very glad, I wish I were there so I could visit Crefeld, but and I show this paper in China a lot just to point out to them that PLOS One paper has more citations than most Nature and Science papers. So just to make sure that China understands the paper matters more than the journal. And then there's this, we of course see this decline. And of course, everybody would like to know what the reasons for the decline are. And is climate change one of those reasons? Okay, so I got incredibly fortunate because Thomas Rosslin and Niels Martin Schmidt invited me to join a consortium group to sequence insects, arthropods that were sampled in Greenland, where humans don't live. And so we can rule out agriculture, we can rule out lots of things, but we cannot rule out climate change as a cause of insect change. And also it's a very simple system, mostly arthropods. And mostly barcoded. And Niels Martin Schmidt, in a genius moment, started this systematic collection using pan pitfall traps in 1996, and has been storing them in alcohol in a warehouse in Denmark since then. And so we were able to get these alcoholic specimens. We estimate about 750,000 individual arthropods in total in the whole thing. And we took one line of traps worth from 1997 to 2013. And we shotgun sequenced each one of them. So we did not use PCR. We just took them, extracted DNA, and just shotgun sequenced them. And so we did something over 700 libraries to do this. So we burned a lot of money to do this. And the reason we did this is we didn't want any effective PCR contamination. So we wanted the number of reads to reflect the amount of biomass. And we were able to get the DNA barcodes and also assemble mitoGenomes for more than 300 species, or partial or full mitoGenomes for more than 300 species. And then what we could do, right, is we could take the raw reads from a sample and just map them to all those mitoGenomes. And so this is a case where this species has lots of reads that map to it and right across the mitogenome. So we can confidently say that this species is present and at high biomass. And then this species there's lots of reads that map to it, but only in that tiny part of the mitogenome. So we say we can rule that out. That's a conserved site, and we can rule out this false positive. And so that turns out to be really, really good at separating false positives from true positives. And so we were able to do some modeling. And then this is what the data set looks like, 1997 to 2013. And then after doing some spiking corrections, we're able to get also within species, like how it's going up and down, right? Blue means 0, no detections. And I think you can see from the left to the right, fewer and fewer colored boxes, right? So fewer and fewer detections as you go from 1997 to 2013. And there's also different sorts of patterns here. So the top 3 here, that's the wolf spider, and then 2 of the fly species. And then you see other ones that show this kind of more fixed activity. And then you see ones that kind of always show up, as occasionally show up, and then they start not showing up. And if you summarize this, what you see is that the species richness in our data set drops by almost 50%. So that's a drop in the species richness right across our whole sample data set, right? Just as the temperature rose on average about 2 degrees, right? So this is like a future time machine for watching the effect of climate change on insect diversity. So we want to know why, right? And again, very, very luckily, Nerea Abrego and also Vaskainen, who, I'll go into them. They literally wrote the book on joint species distribution models, but let me just point out, let's go into a little bit closer. do we see a climate shift, right? That climate correlative approach. So here's a species with the blue that shows an activity peak, a biomass peak in August. And when you go to 2013, it's not active in August anymore. But okay, it's active in July. So that kind of makes sense. Problem is there are other species that also show peaks in August and just don't show up anymore. They just disappear, right? And this is true for several species. They show some kind of activity peak in 1997, and they show no more activity peak when you go to 2013. They're kind of gone. And you see the opposite. You see species that didn't show any activity peak, very rare in 1997, but now are much more abundant in 2013. So we have species going up and species going down. But as you see, in general, species were going down. And so we want to know, can you figure out why that is, right? And so Nerea Abrego and Osa Vizcaíno literally wrote the book on joint species distribution modeling. And they put together this model, right? Species presence or absence in a week, in a year, dependent on the temperature of the previous month, dependent on the week number and the week number squared, so to get the activity peak, and dependent on previous year's own species abundance. That's just all the detections. Previous year's other herbivore abundance, previous year other parasitoid abundance, predators and detritivores. So these are terms that may or may not be significant, and if they're significant, they would be some measure of species interaction. Okay, and did this for 52 species times 542 track weeks, right? And asked how well did the model fit. And they made two models, one with just climate variables in there, and one with climate and the other species in there. So one with species interactions and one with just abiotic interactions. And what they found was that if you had the abiotic model, the model was better than random at explaining the presence and absence of species for all but 4 of the species. So if 4 of the species have an explanatory power less than 0 here, this is a measure of the explanatory power. And so that's worse than random, it's less than 0 here. The interactions model, the one with more terms, they have all of the species are better than random. In fact, you can see that they're just generally better, and their explanatory power is better than the simple abiotic model. So the model fits better if you add other species into the model. Okay, but you could say that they're just more terms, and so they're just overfit, right? So the real test is to fit the model to some of the data and then see if it explains what 1 year that you hold out and you don't use that to fit the model. So that's predictive power. And in general, it's much harder. You get worse results, but you can also see the same effect. So for the abiotic model, the one with just climate variables, and they tried different ones as well, it's better than random for 20% of the species. The abiotic model is worse than random for 80% of the species. So it's terrible at predicting how a species will respond to that climate change over 14 years. And the interactions model is better than random for half the species, which is much, much better. And so this is not overfit because of this training test split. So we would conclude, right, that if you want to predict a species response to climate change, you probably need to know something about the other species in the community. And I think this is a seriously difficult message for predicting the response of biodiversity to climate change. And maybe what's going on with climate change is that it's driving changes in species interactions because it changes the timing, and then that timing changes who interacts with whom. And I think the other key thing is that this was all doable from species for which we have not that much information about their actual what's going on with them. A lot of their larvae interact and do things underground that we don't know anything about, and yet you're still able to pick this up. This is what's a community time series. I think this is the kind of data set that we're going to see and need a lot more of in the future. Okay, fourth topic, the final topic: careers, employment, institutional innovation. I'm a huge Marvel Comics fan, so and this is one of the best television shows that's ever been made, so you should go watch it. Okay, so if you're training, so if you're still a student, postdoc, you can think of different things you might want to learn in conservation and environmental management, and I put them in 5 different boxes. And I think it's realistic to say that no person can be good at all 5 of these, but one person can be good at like 2 of them, right? And I consider myself like half good at lab, half good at analysis, and pretty good at field work. I don't know anything about law, and I certainly don't know anything about funding and scaling and running big organizations. But you can work well with people in the other boxes. So if you want to do conservation and really do this at the scale that it needs to be done, we have to link them all together. And so we have this big circle, the way I think about it. And what we're trying to do is to create as many of these circles as possible. So that's designing an institution. So an institution is a set of rules that get a bunch of people to work together to some common goal. And you assume that they're selfish, but somehow it gets it to work. A university is an institution. And so the nature-space partnership, which I described before, that's a new kind of institution. They invested in that survey that I said, 600 ponds. That map, this is the price list, right? So if you're a builder and you're building in a green zone, you pay somewhere between 0 and 50,000 pounds fee, depending on exactly how large your development is and so on. And then you can do this any time of the year. And then that money goes to pay for new ponds. If you build in the red zone, you pay somewhere between 0 and 100,000 pounds. It depends again on the size of the building. So Nature-Space Partnership created a whole new business on the back of this idea. And you can think of this, each piece of this is different institutions. So Natural England, that's the UK government's, or the English government's Nature Protection Agency, they created this law, right, to protect the great crested newt. And then they also made some changes to allow this eDNA sampling and also to allow this kind of offsetting. So there's a set of laws and rules that they made. And then there was a technological innovation, which is eDNA, to make it possible to survey 600 ponds using eDNA. Never possible with visual surveys. And so Nature-Space Partnership, they got some money from investors, and then Nature-Metrics existed so they could do the 600 samples. Then they used that to parameterize the species distribution model, and then they did funding and scaling, right? They raised private money. They are now raised money to do this, and they're building out and they're working their way across England, Natural England as well. The government is also doing this. Thousands of ponds have been created. Nature-Space by itself has raised £2 million in fees so far. They've created 120 ponds and woodland, and then they also made a management fund that will pay for the management of the ponds going into the future. All this is private sector money paid for directly by the building industry. It never existed before, and then they created this whole new institution. All made possible, right, by technology. So let's go to that tweet, Ben Evans. The first step when you get a new technology is you just do the old thing, but faster, right? So before you wanted to build, there's a pond nearby, you go and collect some water, and then you do that instead of 4 visual surveys. Great, it's faster. But you still have to limit it to 3 months a year, and you still have to wait to see what happens to that pond. And if you get a positive, then you have to do all this translocation, and it's a year's delay and so on. But then what happens is we get to understand the technology. You start to do the same kind of thing, but in a totally different way, right? You get the same outcome, but in a totally different way. So we change the work, right? So Nature-Space replaced surveys and replaced translocations, and they created a situation where you just create new ponds instead, and that removes the risk from construction. So now you have big construction companies, roads, for example, and they just do 100 ponds, and they just see, okay, where are we going to see positives, and then where can we offset that, right? And so I think the way to think about this, and we see this all the time outside of conservation, but inside conservation, we don't really see this as much. The benefit of technology, right, is to change the way we organize, to change the way we run conservation institutions. So that's just a small example of it, but it's really the way we change it. Step 2 is more important than step 1. Okay, and so knowing this, because I'm also interested in tech, I worked for IBM with my first job. We founded Nature-Metrics several years ago. So that's Kat Bruce, she was my PhD student, and Alfie Vogler, who's from Krefeld. And we realized that we tried to do a consultancy model and we weren't going to be able to do that. So we got a little bit of UK money. And then Kat was the CEO for 5 years, and she's now Chief Technical Officer. And we got another round of funding last year for what's called our third growth phase, which we're going to go really big, and we now have something like 60 employees. It's changing all the time. And I think we have probably closer to 30 PhDs at this point. And we're hiring from all over the world, trying to basically stay in one time zone though. You can see we work with NGOs, with large corporations, and so on. We have a couple hundred client organizations, lots of conservation organizations and universities use our services as well, 20,000 samples. We're now building out a whole new laboratory, whole new offices, and so on. We also recently launched eBioAtlas, which is with IUCN. So IUCN will run a funding model to ask corporations to permanently fund eDNA sampling in freshwater around the world, to start building a around the world updatable map of biodiversity in freshwater biodiversity from around the world. So using new kinds of a funding model. So this is the funding and scaling bit as well. If you're interested in freshwater biodiversity, please have a look at eBioAtlas. There's the possibility, if you're working in a really interesting place, that starting next year we might be able to fund eDNA sampling for you. Okay, so I started with Marvel Comics, and of course you might recognize this person, Robert Downey Jr. Turns out that he's not only an actor, he also runs an environmental charity. And so his own money, they donated 50,000 pounds to be the first donor to eBioAtlas. Here he is doing a video webinar with Katie, our CEO, Katie Critchlow, and Kat Bruce, our CTO. Okay, thank you very much.